Welcome to another episode of Catch This. My name is Trudy Ray. SARS-CoV-2 has now been plaguing this planet for a full year. Yet, throughout the whole pandemic, one statistic continues to hold true. Children don't get as sick from this virus, and when they do, they mostly experience mild symptoms, respond well to treatment, recover more quickly than adults, and they have a better prognosis. Why is that? Small children are usually highly susceptible to respiratory infections, as any parent knows. Are children less susceptible to infection with SARS-CoV-2, or are they just less likely to develop clinical symptoms? We still don't have one clear answer to this question, but there are several hypotheses and some limited data. I will discuss those in this video. I'd like to start with the simplest and most obvious hypothesis for why children don't get as sick from SARS-CoV-2 infection. First of all, children are thought to have fewer underlying disorders such as cardiovascular disease, lung disease, or weak immune systems than older adults. And we know that these issues are fairly prevalent in severe COVID-19. Children also have healthier respiratory tracts because of less exposure to cigarette smoke and air pollution. There is also speculation that the non-specific, innate immune response that occurs upon an initial encounter with a pathogen is stronger in children. This type of immune response seems to be delayed in the elderly, and in an effort to catch up, may result in excessive inflammation, thereby ultimately causing more severe damage. As discussed in a previous video, linked here, severe COVID-19 disease is caused by an aberrant, excessive inflammatory response. Another possible explanation is tied to angiotensin-converting enzyme 2, also known as ACE2, the host cell surface protein that serves as a receptor for SARS-CoV-2 entry into cells. ACE2 is prevalent on lung, kidney, intestinal, and arterial cells, where it normally controls blood pressure by regulating the volume of fluids in the body. ACE2 is also an important regulator of the immune response, especially in the context of inflammation. Some suggest that ACE2 is less mature in young children and thus may not function properly as a receptor for SARS-CoV-2. ACE2 is also more abundant on cells of the lower respiratory tract, which is typically the site of severe COVID-19 disease. Consistent with this observation, there are data that indicate that children experience more SARS-CoV-2 infections in the upper respiratory tract than the lower respiratory tract. It has also been suggested that ACE2 is expressed more abundantly on senescent cells. Senescent cells are cells that have stopped dividing and in which DNA replication no longer occurs, usually in response to several stimuli. These types of cells exist predominantly in the elderly. However, senescent cells are still metabolically active and contain all the factors necessary for virus replication. But do children actually become infected with SARS-CoV-2? The data on this topic are somewhat confusing. There are some that seem to show that children under 10 don't become infected as often. However, these findings do not align with results from inquiries assessing viral load in children. After analyzing 3,303 COVID-19 patient sputum samples using two different PCR systems, German virologist Christian Drosten determined that children under the age of 19 produce virtually the same average levels of viral RNA as adults. Drosten argues that studies showing lower viral RNA levels in children relative to adults are subject to sampling bias because children are less likely to have symptoms and are therefore unlikely to be tested early in infection when virus levels in the nasopharynx are high. Instead, children are usually tested as contacts of index or first identified cases in symptom-triggered household studies, meaning that samples from children are obtained later in infection when virus levels in the nasopharynx are reduced. One way of validating this argument would be by analyzing children's stool samples, because viral RNA has been observed in stool samples as well. Consistent with this suggestion, a report in Emerging Infectious Diseases suggests that both mildly symptomatic and asymptomatic children have high levels of viral RNA in the nose and saliva early during infection, but that these levels decline drastically within 1 to 2 weeks. In contrast, Viral RNA levels in the feces remain high for more than 3 weeks after onset of symptoms. All things considered, it is possible that early childhood vaccines provide some protective immunity against SARS-CoV-2. For example, the measles, mumps, and rubella, or the MMR vaccine, could induce antibodies that are broadly neutralizing or inactivating against a number of pathogens, including SARS-CoV-2. There is a hypothesis based on a 30 amino acid sequence similarity between the SARS-CoV-2 spike protein and the measles virus fusion protein, and between the SARS-CoV-2 spike protein and the rubella virus envelope protein. Computational analysis of this region revealed that the sequence is an antigenic epitope in both measles virus and rubella virus. So it is possible that humoral immunity, basically antibodies, induced by MMR vaccination provides children with protection against SARS-CoV-2 infection as well. But of course, at this point, it is merely a hypothesis, which needs to be validated with experimental data. However, there are other observations that have been made in the realm of childhood vaccinations and protection from other infections. According to the Associate Director for Research at the FDA Office of Vaccines Research and Review, Kostya Chumakov, on episode 604 of This Week in Virology, there are viruses that are persistently present in the population, and such viruses can always be isolated from healthy children. Back in the polio days, when they were conducting clinical trials for the oral polio vaccine, also known as OPV, it was found that these other viruses were completely wiped out in children who had received OPV. In fact, an initial administration of OPV reduced mortality caused by other pathogens by as much as 30%. And each successive OPV booster increased this favorable effect even further. This was observed in several parts of the world because it stopped outbreaks caused by completely unrelated viruses. The same effect has also been observed after vaccination with other live attenuated vaccines like the smallpox vaccine and bacterial vaccines like BCG and others. The effect would most definitely have to be non-specific, which implicates the innate immune response. It may just be possible that recent vaccination simply engages the innate immune response and keeps it on guard. And it also makes sense that because immunity derived from these childhood vaccines typically wanes with age, that this waning immunity could increase the risk of severe COVID-19 in the elderly. And finally, the apparent resistance of children to severe COVID-19 may have something to do with the other coronaviruses, the seasonal coronaviruses, NL63, 229E, HKU1, and OC43. Most children produce antibodies to these viruses by the age of 6, so it is possible that these antibodies could protect children from SARS-CoV-2 as well. Children are more likely to have had a recent primary infection with these viruses than adults, so the immune memory may be stronger in children. As is typical of newly emerging pathogens, many characteristics of the diseases they cause are largely unknown. As such, the exact reasons for why COVID-19 is less severe in children remain ambiguous. But hopefully the answer will become clearer as more data emerge. That's it for today. My name is Trudy Ray. I'll see you next time on Catch This.